Good morning, children. Today we'll start with the third lesson, that is chapter 8 of biology. How do organisms reproduce? So far, we have studied two lessons. One is on life processes, and the second one is control and coordination. Right? From those two chapters, we came to know that life processes and control and coordination are very important for our survival. Without those two, if life processes some stops, our life will not be a normal. Even control and coordination will be almost dead. Without those two functions, we won't be able to survive, or we can't lead our normal life. Whereas this reproduction, it's not so important as those two chapters. We can survive without reproduction. We can lead our life normally without reproduction. So, why should we study reproduction? What is reproduction and why should we study reproduction? Let us see in this chapter why reproduction is so important and what is the purpose of reproduction. Reproduction, as such, it is a biological process of all the living organisms. Reproduction means giving birth to young ones of the same species. Here are the four photographs. Here, human beings give birth to human beings, and the plant gives birth to fruits, and which will in turn leads to growth of the plant again. This is a microorganism, bacteria growing from the parent to the offspring, and similarly, animals also will reproduce the same kind. This is a simple definition of reproduction, that is, giving birth to young ones. Now, let us see what is the purpose of reproduction. Why? Is it necessary? Reproduction, as such, we have just now we heard that reproduction is not compulsory, necessary. It's not compulsory process. If reproduction doesn't happen, what will happen? If we don't reproduce, what will happen to the earth or life? See, these are the species where dinosaur and long tusked elephant or Tasmanian tiger, these animals, they have become extinct. Now we cannot see these animals. Okay? Why? Because of the loss in reproduction, these animals become extinct. Have become extinct. Extinct and there doesn't exist after that. That does not exist. So even if we also, all the living beings, if we don't reproduce, population of the species reduces over time. Population itself shrinks and it becomes endangered. Finally, it becomes extinct. So we have to reproduce. All living beings have to reproduce to continue our life, continue the life on earth. To have a continuous life on earth, reproduction is very much necessary. Okay, what is reproduction? We know giving birth to young ones. During the process, what happens? In earlier classes and all, last year you have studied DNA is the genetic material that passes from one generation to another generation. During reproduction also, DNA gets copied from parents to offspring. Why it is important? Why DNA should be copied from parents to offspring or parents to younger ones? Because information of the characters. Say parents to characters, it will pass from parents to offspring through DNA, because information of those characters are stored in the form of DNA. So information of characters passes from one generation to another generation through DNA. By passing it, DNA, it gets converted to proteins. Proteins which decides the structure of the body. It is not information is there in the DNA which leads to formation of proteins. Because of these proteins, we get a defined body structure. If there is any error in DNA, protein will get altered, so our body structure also gets altered, right? Thirdly, because of this variation in error in DNA, variation in protein structure, and variation in body structure, there will be variation among individuals. For example, if one parent has 4 children, 4 daughters or 4 boys, sons, we can see some kind of variation within that 4 kids, right? Because though DNA copying looks similar, but it is not exact as its original. So it creates variation. So reproduction is important to continue our life on earth and pass information from parents to offsprings. Thirdly, to have a defined body structure. And finally, to have a variation among individuals. Why variation is necessary? This we know, due to this copying, during the process of copying of DNA from parents to offspring, there creates a variation. Why variation is necessary during reproduction? Why should we have variation among ourselves? See, this is a evolution of picture showing evolution of human beings. We know that we have evolved. Now this is a human being, what we are seeing. We have seen that we are evolved from a ape or chimpanzee like animals, and with millions of years we have evolved like this because of the variation. Why should we require? We know the environment at this age was totally different from this age. Because of the variation among animals, though there is a drastic change in the environment, some of the individuals get survived in the extreme conditions. So they'll survive with the drastic change in the environment and they'll move on with modifications. Similarly, with change in environment or any other changes, resistant species get acclimatized and they change accordingly and their life proceeds. Okay, and it also, this variation, because of this variation, the species will not become extinct. Okay, why variation is necessary? Because we need evolution. Different forms will be changed. Variation is necessary for evolution of living beings into with new forms and acclimatization of the new environment, drastic change in the environment, and we don't become extinct. These are the 3 reasons why variation is necessary. From these, we know reproduction is very much necessary, though it is not very much required as life processes and control and coordination, but this is also necessary to have a life on earth. Okay, for example, I'll give you why variation is necessary. ಈಗ ನೋಡ್ತಾ ಇದೀವಿ, ಎಲ್ಲ ಕಡೆ ಕೊರೋನಾ ವೈರಸ್ ಇನ್ಫೆಕ್ಷನ್ ಸ್ಪ್ರೆಡ್ ಆಗ್ತದೆ. ಎಷ್ಟೋ ದೇಶಗಳಲ್ಲಿ ಎಷ್ಟೊಂದು ಜನ ಸಾಯ್ತಾ ಇದ್ದಾರೆ. ಅಲ್ಲಿನೂ ಒಬ್ಬರಿಂದ ಒಬ್ಬರಿಗೆ ವೇರಿಯೇಷನ್ ಇರೋದ್ರಿಂದ ಯಾರು ಸಸೆಪ್ಟಬಲ್ ಅವ್ರಿಗೆ ಇನ್ಫೆಕ್ಷನ್ ಆಗಿ ಅವರು ದೇ ಆರ್ ಬೀಯಿಂಗ್ ಡೆಡ್. Whereas the individuals who have strong immunity because of variation in the genetics information, they fight against that virus. They are fighting against the ಆ ವೈರಸ್ನ ಫೈಟ್ ಮಾಡ್ತಾ ಇದ್ದಾರೆ and they are able to survive and move on. This is a simple example why variation is necessary during reproduction. Okay, now with this importance of reproduction, and we know why reproduction is necessary, let us study different methods of reproduction in living organisms. There are two types of reproduction. One is sexual reproduction and another is asexual reproduction. Sexual reproduction requires two individuals of different sex. Female and female parent is required for sexual reproduction. Whereas asexual reproduction, it requires only one parent. There is no involvement of sex cells. Sex cells iralla. Eitherly, only one parent is used for asexual reproduction. This is a chart showing different forms of asexual reproduction, like binary fission, multiple fission, budding, fragmentation, sporulation, and in plants, vegetative propagation. Let us see where all these, in which kind of organisms this type of asexual reproduction occurs. Now let us see asexual reproduction in unicellular organism. You know, unicellular organism is the organism having only one cell. In that, in these unicellular organisms, two kinds of asexual reproduction happens. One is binary fission and another is multiple fission. What is binary fission? Here you can see the picture of amoeba. Amoeba, we know, it doesn't have a definite shape. During the growth of the amoeba, sometimes nucleus, it gets divided into two. And so, along with the nucleus, cellular contents also divide into two equal halves and becomes two individuals. This is called binary fission. It's just a division of nucleus and the cellular contents into two. This division can occur at any plane. It can occur here or it can happen here. Any angle division can happen. This is called binary fission in amoeba. Now, in another unicellular organisms called leishmania, it is a parasite which caused a disease called Kala Azar in human beings. Kala Azar andre, it's a very scary disease where skin starts turning black and will become weak, liver bulges. It's a very scary disease. That parasite inside our body, this is the structure of the parasite by leishmania. Here, division occurs at a particular orientation, fixed orientation. Here, the tip of the organism divides and division occurs only at this angle. It cannot happen at any angle. This is, division is not fixed, whereas here division happens at a fixed orientation. Here also the division is binary fission and it divides into two equal cells, new cells. Okay. Whereas in multiple fission, this is a cell of a plasmodium. Plasmodium is a malarial parasite that induces malaria. When the mosquito plasmodium enters into our body, here within the cell, the nucleus and the cellular contents undergo multiple fissions, multiple cells, and after at a particular favorable condition, this cell wall breaks, cyst breaks, and the daughter cells will be released into our body. Thus, we get infected with malaria. If it were to be binary fission, the infection wouldn't have been so fast. Since it is multiple fission, infection of malaria in human body drastically increases. This is called multiple fission. Means single cell along with the nucleus, it divides into many daughter cells. That is called multiple fission. Binary in a bi and the two, it divides into two cells, whereas multiple means many cells. For example, Plasmodium has multiple fission, whereas amoeba and Leishmania has binary fissions. This is with respect to unicellular organisms. Now let us see what happens, how asexual reproduction happens in multicellular organisms. Multicellular organisms, there are four different types of asexual reproduction. One is fragmentation, regeneration, budding, and spore formation. Let us see what is fragmentation. For example, in Spirogyra, what happens? This is a Spirogyra filament. If due to unfavorable conditions, it breaks. It becomes fragmented. And when the fragment happens, this Spirogyra will never die. Each fragment will develop into a new cell, new Spirogyra. And so we'll have two offsprings, two new individuals will be formed. It can break up into any pieces. Each fragment will become a new cell. This is called fragmentation. This is a fragment and this is a fragment. If it cuts here, again, it grows into a new individual. Whereas regeneration, during, for example, planaria and hydra, it can cut. They can be cut at different, any number of cuts can happen. Each cut, say, this is a top portion and this is a lower portion. If this is the individual, the lower portion gets regenerated, whereas with the lower portion, upper portion gets regenerated. Similarly, in case of hydra, ಈಗ ಎಕ್ಸಾಂಪಲ್ ಏನು ಕೇಳಿದ್ದೀನಿ, ಈಗ ಹಲ್ಲಿನ ನಾವು ಅಕಸ್ಮಾತ್ ಮನೆಯಲ್ಲಿ ಹಲ್ಲಿನ ಹೊಡೆದ್ರೆ ಅದು ಫಸ್ಟ್ ಆದ್ರೂ ಬಾಲ ಕಟ್ಟಾಗತ್ತೆ, ಹೌದಾ? ಬಾಲ ಕಟ್ಟಾದ್ರೆ ಏನಾಗತ್ತೆ, ಆಫ್ಟರ್ ಸಮ್ ಟೈಮ್ ಬಾಲ ಮತ್ತೆ ಬೆಳೆಯತ್ತೆ ಆ ಹಲ್ಲಿಯಲ್ಲಿ. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ರೀಜನರೇಷನ್. Similarly, in case of hydra and planaria, each cut portion will develop into fully developed individuals. That will become a new organism. This is with respect to, so this is called regen. The remaining portion regenerates. That's why this process is called regeneration. Now, budding. Hydra undergoes two forms of asexual reproduction. One is regeneration and another is budding. What is budding? You can see in this diagram, there will be a small outgrowth in the structure of hydra. This due to division, maybe due to division of serial cell division at a particular site. Because of this outgrowth, it develops into a bud. A bud form agatte. After certain stage, when this bud becomes mature, it separates from the mother plant. It separates from the mother and becomes an independent individual. First, there will be a small outgrowth. This outgrowth will grow into a bud. After maturity, this becomes separated from the mother and becomes a separate individual. This will be in this type of reproduction we can see in hydra and also in yeast. Okay, yeast is a single unicellular organism where budding can be seen in yeast, whereas hydra is a multicellular organism. Now coming to a fourth type of asexual reproduction in multicellular organisms, that is spore formation. Spore formation ಅಂದ್ರೆ ಏನು? ಈಗ ಅಕಸ್ಮಾತ್ ಬ್ರೆಡ್ ಮೇಲೆ ನೀವು ಫಂಗಸ್ ಗ್ರೋತ್ ಆಗುತ್ತೆ ನೋಡಿದ್ದೀರಾ? ಅಲ್ಲಿ ದಾರದ ತರ ಥ್ರೆಡ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಇರುತ್ತೆ. ದೀಸ್ ಥ್ರೆಡ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ಸ್ ಆರ್ ಕಾಲ್ಡ್ ಐಫಾ. ಓಕೆ, ಐಫಾ ಅಂತ ಕರಿತೀವಿ. ಒಂಥರ ದಾರ ತರ ಇರುತ್ತೆ. ಅಕಸ್ಮಾತ್ ನೀವು ಒಂದು ಬಾಕ್ಸ್ ಅಲ್ಲಿ ಏನಾದ್ರು ಬ್ರೆಡ್ ಮುಚ್ಚಿಟ್ಬಿಟ್ಟು ಆಫ್ಟರ್ ಸಮ್ ಟೈಮ್ ಸ್ವಲ್ಪ ದಿವಸ ಆದ್ಮೇಲೆ ತೆಗೆದ್ರೆ ಸಡನ್ ಆಗಿ ಸ್ಪೋರ್ಸ್ ಒಂಥರ ಗಾಳಿಗೆ ಏನೋ ಬಂದಂಗ ಆಗುತ್ತೆ. ದೋಸ್ ಆರ್ ಕಾಲ್ಡ್ ಸ್ಪೋರ್ಸ್. ಎಲ್ಲಿ ಫಾರ್ಮ್ ಆಗುತ್ತೆ? ಹೆಂಗ್ ಸ್ಪೋರ್ಸ್? ಈ ಐಫಾ ತುದಿಯಲ್ಲಿ ದೇರ್ ಬಿ ಬಲ್ಬ್ ಲೈಕ್ ಸ್ಟ್ರಕ್ಚರ್ಸ್, ಅಂದ್ರೆ ಟೈನಿ ಬಲ್ಬ್ ಸ್ಟ್ರಕ್ಚರ್ ಇರುತ್ತೆ. ಅದು ಇಟ್ ವಿಲ್ ಬಿ ವೆರಿ ಹಾರ್ಡ್. ಅದ್ರೊಳ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಸ್ಪೊರಾಂಜಿಯಾ. ಸ್ಪೊರಾಂಜಿಯಾ ಅಂತ ಕರಿತೀವಿ. ದಿಸ್ ಸ್ಪೊರಾಂಜಿಯಾ ಒಳಗೆ ಸ್ಮಾಲ್ ಡಸ್ಟ್ ಪಾರ್ಟಿಕಲ್ಸ್ ತರ ಸ್ಪೋರ್ಸ್, ಸ್ಪೊರಾಂಜಿಯಾ ಒಳಗೆ ಸ್ಪೋರ್ಸ್ ಇರುತ್ತೆ. ಇದು ಯಾವಾಗ್ಲೂ ಓಪನ್ ಆಗಲ್ಲ. ಓನ್ಲಿ ಇಫ್ ಮೋಲ್ಡ್ಸ್ ಯಾವಾಗ ಫಾರ್ಮ್ ಆಗುತ್ತೆ? ಒಂದು ದೇರ್ ಇಸ್ ಅ ವೆಟ್, ಅಂದ್ರೆ ಮಾಯಿಸ್ಟ್ ಇದ್ದಾಗ mold form ಆಗುತ್ತೆ. These sporangia burst open and spores will come out and they grow into a fungus when moist surface is there. ಅಂದ್ರೆ ಒದ್ದೆ ಇದ್ದಾಗ ಈ ಸ್ಪೊರಾಂಜಿಯಾ ಓಪನ್ ಆಗಿ ಸ್ಪೋರ್ಸ್ ಆಚೆಗೆ ಬಂದು ಇಟ್ ಗ್ರೋಸ್ into a different fungus. For example, rhizopus, it's a bread mold. ಬ್ರೆಡ್ ಮೇಲೆ ದಾರದ ತರ ಐಫಾ ಇರುತ್ತೆ. ಐಫಾ ತುದಿಯಲ್ಲಿ ಸ್ಮಾಲ್ ಬಡ್ಸ್ ತರ ಇರುತ್ತೆ. ಅದ್ರೊಳಗೆ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಸ್ಪೊರಾಂಜಿಯಾ. ದಟ್ ವಿಲ್ ಬಿ ವೆರಿ ಥಿಕ್. ಅದು ಯಾವಾಗ ಫೇವರಬಲ್ ಕಂಡೀಷನ್ಸ್ ಲೈಕ್ ಮಾಯಿಶ್ಚರು, ಏರ್ ಕಮ್ಮಿ ಇದ್ರೆ ದಿ ಸ್ಪೊರಾಂಜಿಯಾ ಓಪನ್ಸ್ ಅಪ್ ಅಂಡ್ ರಿಲೀಸಸ್ ಸ್ಪೋರ್ಸ್. ಸ್ಪೋರ್ಸ್ ಆಚೆಗೆ ಬಂದು ಫಂಗಸ್ ಗ್ರೋತ್ಸ್ ಆನ್ ದ ಬ್ರೆಡ್. This type of asexual reproduction is called spore formation. Now we know there are 4 different types of asexual reproduction in multicellular organisms. Now after knowing all this, now let us see vegetative propagation in plants. Vegetative or asexual reproduction in plants. In plants, new plants will be produced from vegetative parts of the original plant. Vegetative parts ಅಂದ್ರೆ ಸ್ಟೆಮ್ಮು, ರೂಟು, ಲೀವ್ಸು, ಅದರಿಂದ ಹೊಸ ಪ್ಲಾಂಟ್ಸ್ ಬರೋದನ್ನ asexual reproduction. ಅದು ಏನಕ್ಕೆ? ವೈ ಅಡ್ವಾಂಟೇಜಸ್ ಆಫ್ ದಿಸ್ asexual reproduction ಏನೇನು? ಒಂದು, ಸೀಡ್ಸ್ ಇಂದ ಬರೋ ಪ್ಲಾಂಟ್, ಸೆಕ್ಷುವಲ್ ರಿಪ್ರೊಡಕ್ಷನ್ ಇಂದ ಬರೋ ಪ್ಲಾಂಟ್ಸ್ ಗಿಂತ ಮುಂಚೆ ಫ್ರೂಟ್ಸ್ ಕೊಡುತ್ತೆ. ಫ್ರೂಟ್ಸ್, ಫ್ಲವರ್ಸ್ ಆರ್ ಫ್ರೂಟ್ಸ್ ಅರ್ಲಿಯರ್, ಅಂದ್ರೆ ಬೇಗ ಕೊಡುತ್ತೆ ಅಂಡ್ ಈಸಿ ಟು ಗ್ರೋ. ಇದೆಲ್ಲ ತುಂಬಾ ಬೇಗ ಈಸಿ ಟು ಗ್ರೋ ಅಂಡ್ ಕಾಸ್ಟ್ ಈಸ್ ಆಲ್ಸೋ ಕಮ್ಮಿ ಆಗುತ್ತೆ. ಅಂಡ್ ಪಾರ್ಥೆನೋಕಾರ್ಪಿಕ್ ಫ್ರೂಟ್ಸ್ ಅಂದ್ರೆ ಸೀಡ್ಲೆಸ್ ಫ್ರೂಟ್ಸ್ ನ ಡೆವಲಪ್ ಮಾಡಕ್ ಹೆಲ್ಪ್ ಮಾಡುತ್ತೆ. ಇದು ಸೀಡ್ಸ್ ಇಂದ ಬರದೇ ಇರೋದ್ರಿಂದ these plants obtained from asexual reproduction are genetically similar. And the genetics, DNA will be almost same. There is no variation in this plants produced from asexual reproduction. ಒಂದು ಬೇಗ ಫ್ರೂಟ್ಸ್ ಅಂಡ್ ಫ್ಲವರ್ಸ್ ಕೊಡುತ್ತೆ and it loses the capacity to produce seeds. ಈಗ ಬನಾನಾ ಅಲ್ಲಿ ನೋಡ್ತಾ ಇದ್ರೆ ಇದು ಬಿಕಾಸ್ ಆಫ್ ದ ವೆಜಿಟೇಟಿವ್ ಪ್ರಾಪಗೇಷನ್ ಸೀಡ್ಸ್ ಸೆಟ್ಟೇ ಆಗಲ್ಲ. ಅದು ಸಿಮಿಲರ್ಲಿ ಪೊಟ್ಯಾಟೊ ಸೀಡ್ ಸೆಟ್ ಆಗದೇ ಇಲ್ಲ. ಆಮೇಲೆ ಸೀಡ್ಲೆಸ್ ಫ್ರೂಟ್ಸ್ ಆಗಿ ಬೇಗ ಮಾಡ್ಬೋದು. ಈ ಮೆಥಡ್ ಅಲ್ಲಿ ಸೀಡ್ಲೆಸ್ ಫ್ರೂಟ್ಸ್ ಗ್ರೋ ಮಾಡ್ಬೋದು. ಸೀಡ್ಲೆಸ್ ಗ್ರೇಪ್ಸ್ ಆಗ್ಬೋದು, ಸೀಡ್ಲೆಸ್ ಬನಾನಾ ಆಗ್ಬೋದು, ಸೀಡ್ಲೆಸ್ ವಾಟ್ ಈಸ್ ದಟ್ ಸ್ವೀಟ್ ಇದಾಗ್ಬೋದು. ಸೀಡ್ಲೆಸ್ ಫ್ರೂಟ್ಸ್ನ ಡೆವಲಪ್ ಮಾಡ್ಬೋದು. ಅಂಡ್ ಇಟ್ ಈಸ್ ಚೀಪರ್ ಅಂಡ್ ಟೈಮ್ ಈಸ್ ವೆರಿ ಲೆಸ್. Easy and cheap method of growing plants. These are the advantages of Asexual reproduction. Now let us see ಸ್ಟೆಮ್ ಇಂದ ಯಾವ್ಯಾವ ಪ್ಲಾಂಟ್ಸ್ ಬರುತ್ತೆ, ಯಾವ್ಯಾವ ಪ್ಲಾಂಟ್ಸ್ ಸ್ಟೆಮ್ ಯೂಸ್ ಮಾಡಿಕೊಂಡು ನ್ಯೂ ಪ್ಲಾಂಟ್ ಫಾರ್ಮ್ ಮಾಡ್ಬೋದು ಅಂತ ನೋಡೋಣ. ಎಕ್ಸಾಂಪಲ್ ಆನಿಯನ್, ಬಲ್ಬ್ಸು, ಬನಾನಾ, ಸಕ್ಕರ್, ರೈಸೋಮ್, ಜಿಂಜರ್, ಟ್ಯೂಬರ್. ಈಗ ಇಲ್ಲಿ ನೋಡೋಣ, ದೀಸ್ ಆರ್ ದ ಬಲ್ಬ್ಸ್. ಅಂದ್ರೆ ಸ್ಟೆಮ್ಸ್ ನ ಡಿಫ್ರೆಂಟ್ ಫಾರ್ಮ್. ಆನಿಯನ್ಸ್ ಅಲ್ಲಿ ಬಲ್ಬ್ಸ್ ಅಂತ ಕರಿತೀವಿ. ದೀಸ್ ಬಲ್ಬ್ಸ್, ಆನಿಯನ್ ಅಲ್ಲಿ ಎಲ್ಲೂ ನೀವು ಸೀಡ್ ಯೂಸ್ ಮಾಡಲ್ಲ ಟು ಹ್ಯಾವ್ ಅ ನ್ಯೂ ಪ್ಲಾಂಟ್. ಬಲ್ಬ್ಸ್ ಯೂಸ್ ಮಾಡ್ತೀವಿ. ಓಕೆ, ಬಲ್ಬ್ಸ್ ಯೂಸ್ ಮಾಡಿ ಫ್ರಮ್ ದಿಸ್ ನ್ಯೂ ಪ್ಲಾಂಟ್ ವಿಲ್ ಅರೈಸ್ ಫ್ರಮ್ ದ new onion plant will arise from this. This is the bulbs. It's a modified stem. Whereas in banana, ಕೆಳಗಡೆ ಬೇಸ್ ಆಫ್ ದ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಸಕ್ಕರ್ಸ್ ಅಂತ ಬರುತ್ತೆ. A new plant will arise. ಇದನ್ನು ಉಪಯೋಗಿಸ್ಕೊಂಡು we can grow a new banana plant and we yield banana, and it takes less time to yield banana. Okay. Whereas ginger, rhizomes, ಇಲ್ಲಿ ಈ ರೂಟ್ಸ್, ಬಡ್ಸ್ ಇಂದ ರೂಟ್ಸ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ, ಅದರಿಂದ ನ್ಯೂ ಪ್ಲಾಂಟ್ಸ್ ಬರುತ್ತೆ. ದಿಸ್ ಇಸ್ ಅ ಮಾಡಿಫೈಡ್ ಸ್ಟೆಮ್ ಆಫ್ ಜಿಂಜರ್ ಇನ್ ದ ಫಾರ್ಮ್ ಆಫ್ ರೈಸೋಮ್. whereas potato, ಟ್ಯೂಬರ್ಸ್, ಇಟ್ಸ್ ಅ ಟ್ಯೂಬರ್, ಅಲ್ಲಿ ಹೈ ಬಡ್ಸ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ. ಫ್ರಮ್ ದಿಸ್ ಹೈ ಬಡ್ಸ್ new plant, when it is put it in the soil, new plant will arise from this high buds. These are the examples of plants obtaining from stems. Now leaves in the ಯಾವ plants ಬರುತ್ತೆ? This is a picture called bryophyllum. Bryophyllum leaf ಇದು. ಆ edges, margin ಅಲ್ಲಿ plantlets, new buds or plantlets ಆಚೆಗೆ ಬರುತ್ತೆ. ಈ ಪ್ಲಾಂಟ್ಲೆಟ್ಸ್ ಏನಾದರೂ ಇದರಿಂದ ಉದ್ರಿ ಕೆಳಗೆ ಬಿದ್ದರೆ, ಸಾಯಿಲ್ ಒಳಗೆ ಬಿದ್ದರೆ, each plantlets will become a new individual. This regeneration or reproduction we can see in bryophyllum. Leaf margin ಅಲ್ಲಿ plantlets ಅಥವಾ buds form ಆಗುತ್ತೆ. If these buds fall into the soil, each plantlet or bud will become a new plant. This starts with the leaf. Now ಅದೇ ತರ ರೂಟ್ಸ್ ಇಂದ ಯಾವುದು? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ಡೇಲಿಯಾ ನೋಡಿದ್ದೀರಾ? ಹೂವು, ಡೇರೆ ಹೂವು ಅಂತ ಹೇಳ್ತೀವಿ. ಅದ್ರ ಗೆಡ್ಡೆ ಕೆಳಗಡೆ ನೆಲದಲ್ಲಿರೋ ಗೆಡ್ಡೆಯಿಂದ, ದಟ್ ಈಸ್ ಅ ರೂಟ್, ಮಾಡಿಫೈಡ್ ರೂಟ್. ಅದರಿಂದ ಹೊಸ ಗಿಡ ಬಂದು ಡೇರೆ ಹೂವು ಬಿಡುತ್ತೆ. ಅದೇ ತರ ಸ್ವೀಟ್ ಪೊಟ್ಯಾಟೊ, ನ್ಯೂ ಪ್ಲಾಂಟ್ಸ್ ಅರೈಸ್ ಫ್ರಮ್ ಅಡ್ವೆಂಟಿಷಿಯಸ್ ರೂಟ್ಸ್. Example, dahlia and sweet potato. This is a natural asexual reproduction. Andre natural form of a plant inda will get a natural propagation. Whereas artificial vegetative propagation, artificial methods of vegetative propagation andre enu? This is not from the any tissue like leaves, stem idrandalla. ಇಲ್ಲಿ ನಾವು ಮಾಡೋದು ವಿ ಕ್ರಿಯೇಟ್ ವೆಜಿಟೇಟಿವ್ ಪ್ರಾಪಗೇಷನ್. ಹೆಂಗೆ? ಸ್ಟೆಮ್ ಕಟ್ಟಿಂಗ್. ಏನ್ ಮಾಡ್ತೀವಿ ನಾವು? ಇವಾಗ ರೋಸ್ ತಗೊಂಡ್ರೆ ಸ್ಮಾಲ್ ಅಂದ್ರೆ ಬಲ್ತಿರೋ ಸ್ಟೆಮ್, ಸ್ವಲ್ಪ ದಪ್ಪ ಇರೋ ಸ್ಟೆಮ್ ಅಲ್ಲಿ ವಿಲ್ ಕಟ್ ದ ಸ್ಟೆಮ್ ಲೈಕ್ ದಿಸ್. ಫಾರ್ ಎಕ್ಸಾಂಪಲ್ ದಿಸ್ ವಿತ್ ನೋಡ್ಸ್ ಅಂಡ್ ಇಂಟರ್ನೋಡ್ಸ್ ಅಂಡ್ ಇಲ್ಲಿ ಬಡ್ಸ್ ಇದೆ, ಇಲ್ಲಿ ನೋಡ್ಸ್, ದಿಸ್ ಇಸ್ ಅ ನೋಡ್, ದಿಸ್ ಇಸ್ ಅ ಇಂಟರ್ನೋಡ್. ಇಫ್ 2 to 3 ಇಂಟರ್ನೋಡ್ಸ್ ಇರಬೇಕು, 2 ಬಡ್ ಇರಬೇಕು, ನೋಡ್ಸ್ ಇರಬೇಕು. ಇದನ್ನ ನಾವೇನಾದ್ರೂ ಸಾಯಿಲ್ ಒಳಗೆ ಪ್ಲಾಂಟ್ ಮಾಡಿದ್ರೆ, ಸಾಯಿಲ್ ಒಳಗೆ ಹಾಕಿದ್ರೆ, ಆಫ್ಟರ್ ಸಮ್ ಟೈಮ್ ಇಫ್ ಯು ಗಿವ್ ಫರ್ಟಿಲೈಸರ್ ಅಂಡ್ ವಾಟರ್ ಎವ್ರಿಥಿಂಗ್, ಆಫ್ಟರ್ ಸಮ್ ಟೈಮ್ ದೀಸ್ ಕಟ್ಟಿಂಗ್ಸ್ will develop a roots and also shoots. New plants will germinate from the stem cutting. This we can see in ಕಬ್ಬು ನೋಡಿದಿರಾ? ಕಬ್ಬನಲ್ಲೂ ಬೀಜ ಹಾಕಲ್ಲ ಅಥವಾ ಇದು ಹಾಕಲ್ಲ, ಅಥವಾ ರೋಸ್. ದೀಸ್ ಥಿಂಗ್ಸ್ ವಿಲ್ ಬಿ ಡನ್ ವಿತ್ ಕಟ್ಟಿಂಗ್, ಸ್ಟೆಮ್ ಕಟ್ಟಿಂಗ್. ಕಬ್ಬು ಮತ್ತೆ ರೋಸು ಸ್ಟೆಮ್ ಕಟ್ಟಿಂಗ್ ಯೂಸ್ ಮಾಡಿ ಮಾಡ್ತಾರೆ. ಸೆಕೆಂಡ್ ಇಸ್ ಲೇಯರಿಂಗ್. ಲೇಯರಿಂಗ್ ಅಂದ್ರೆ ಏನು? ವಾಟ್ ವಿ ಡೂ, ದಿಸ್ ಇಸ್ ಅ ಒನ್ ಪ್ಲಾಂಟ್, ವಿಲ್ ಬೆಂಡ್ ದ ಲೋಯೆಸ್ಟ್ ಸ್ಟೆಮ್ ಇದನ್ನ ಬೆಂಡ್ ಮಾಡಿ ಸಾಯಿಲ್ ಒಳಗೆ ಬರಿ ಮಾಡ್ತೀವಿ. ಬೈ ಲಿವಿಂಗ್ ದ ಟಿಪ್, ಗ್ರೋಯಿಂಗ್ ಟಿಪ್ನ ಆಚೆಗೆ ಬಿಟ್ಟು ಉಳಿದಿರೋ ಸ್ಟೆಮ್ನ ಸಾಯಿಲ್ ಒಳಗೆ ಮುಚ್ತೇವೆ. ಹಂಗೆ ಮುಚ್ಚಿದಾಗ ಇಯರ್ ದ ರೂಟ್ ಸ್ಟಾರ್ಟ್ಸ್ ಗ್ರೋಯಿಂಗ್ ಅಂಡ್ ಶೂಟ್ ಆಲ್ಸೋ ಸ್ಟಾರ್ಟ್ಸ್ ಗ್ರೋಯಿಂಗ್. ಇದು ರೂಟ್ ಫಾರ್ಮೇಶನ್ ಆದಾಗ ವಿ ಕ್ಯಾನ್ ಕಟ್ ಇಟ್ ಇಯರ್ and we can plant this into a new plant. This again it becomes a new plant. ಎಲ್ಲಿ ಮಾಡ್ತೀವಿ ಇದನ್ನ? ಮಲ್ಗೆ ಗಿಡ, ಈಗ ಒಂದ್ ಮಲ್ಗೆ ಗಿಡ ಇರುತ್ತೆ ನಿಮ್ಮ ಮನೇಲಿ, ನಾನು ಒಂದ್ ಮಲ್ಗೆ ಗಿಡ ಕೊಡಿ ಅಂತ ಕೇಳ್ತೀವಿ. ಅವಾಗ ಹೆಂಗ್ ಮಾಡ್ಬೇಕು ನೀವು? ಈ ತರ ಲೇಯರಿಂಗ್, ಒಂದು ಸ್ಟೆಮ್ನ ಸಾಯಿಲ್ ಒಳಗೆ ಬರಿ ಮಾಡಿದ್ರೆ ರೂಟ್ ಫಾರ್ಮ್ ಆಗುತ್ತೆ, ಶೂಟು ಗ್ರೋ ಆಗುತ್ತೆ. ಅವಾಗ ಯು ವಿಲ್ ಕಟ್ ದಿಸ್ and you can give this entire plant to me. Okay, this is done by layering. Okay, jasmine and strawberry can be grown with this type of vegetative propagation. Third type is grafting. Grafting andre enu? It is done with the two plants, two individuals. Whereas idrallela one stem, one cutting ತಗೊಂಡ್ವಿ, ಒಂದೇ plant ಇಂದ ಇದು ಮಾಡಿದ್ವಿ. Whereas grafting ಏನಂದ್ರೆ this root portion is called stalk. This most of the times it will be wild variety stalk irutte. Wild variety yaake andre it is resistant to most of the pathogens athwa diverse adverse climatic and stresses ಗೆ resistant irutte anta usually stalk will be from wild variety and it will be thick. And stem portion is called scion, root portion is called stalk. ಇದನ್ನ ಬೇರೆ ಪ್ಲಾಂಟ್ ಇಂದ ತಗೋತೀವಿ ಸಯಾನ್ ಅಂತ. ಮತ್ತೆ ಅದೆರಡನ್ನು ವಿ ಹ್ಯಾವ್ ಟು ಕಟ್ ಆಬ್ಲಿಕ್ಲಿ, ಅಂದ್ರೆ ಈಕ್ವಲ್ ಶೇಪ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡಬೇಕು. ಸೊ ದಟ್ when scion is placed on the stalk it should ಕರೆಕ್ಟಾಗಿ ಫಿಕ್ಸ್ ಆಗಬೇಕು. ಆ ತರ ಕಟ್ ಮಾಡ್ಬೇಕು ನಾವು. ಒಂಚೂರು ಗ್ಯಾಪ್ ಇದ್ರೂ ಓಕೆ, ಸ್ವಲ್ಪ ಗ್ಯಾಪ್ ಇದ್ರೂ ಓಕೆ, ಬಟ್ see to it should fix exactly. ಆ ತರ ಕಟ್ ಮಾಡಿ we have to fix this. ಈ ತರ ಒಂದ್ರ ಮೇಲೆ ಒಂದು ಕೂರ್ಸಿದ ತಕ್ಷಣ we'll tie this with the thread, ಮಾಯಿಶ್ಚರ್ ಫರ್ಟಿಲೈಸರ್ ಹಾಕ್ಬಿಟ್ಟು ಸ್ವಲ್ಪ ಗೊಬ್ಬರ ಹಾಕ್ಬಿಟ್ಟು ವಿಲ್ ಟೈ ಇಟ್. ಈ ತರ ಟೈ ಮಾಡಿದಾಗ that will unite, these two pieces gets united and they'll grow into a new plant. Okay, this is called grafting. Usually it is done with grapes and mango. Mango ಏನ್ ಮಾಡ್ತೀವಿ, ಒಂದ್ ಯಾವ್ದೋ ವೆರೈಟಿ ಇರ್ತೀವಿ, ಅವಾಗ ಏನ್ ಮಾಡ್ತೀವಿ, ಒಂದು ಶೂಟ್ನ ಆಲ್ಫಾನ್ಸೋ ಬಾದಾಮಿಯಿಂದ ಗ್ರಾಫ್ಟ್ ಮಾಡ್ತೀವಿ, ಇನ್ನೊಂದು ಟ್ವಿಗ್ಗಿಗೆ ರಸ್ಪುರಿಯಿಂದ ಗ್ರಾಫ್ಟ್ ಮಾಡ್ತೀವಿ, ಇನ್ನೊಂದು ಮೂರನೇ ಟ್ವಿಗ್ಗಿಗೆ ಇಫ್ ವಿ ಗ್ರಾಫ್ಟ್ ಮಲ್ಲಿಕಾ, we'll get 3 varieties in the same plant. One twig, one scion in the mallika barutte, one scion in the badami barutte, one scion in the raspberry barutte. This is the advantage of grafting. We can have more different species in from a single stalk, but scion will be different. Scion will be from 3 different plants, where the stalk is same. Okay, this is useful for grapes and mango. Fourth is tissue culture. Tissue culture andre it is done under controlled condition, ಅಂದ್ರೆ laboratory conditions ಅಲ್ಲಿ ಡೆವಲಪ್ ಮಾಡ್ತೀವಿ tissue culture. How will we do? We'll take a tissue of a plant, either a leaf tissue ಅಥವಾ embryo, ಏನೋ ಒಂದು ತಗೊಳ್ತೀವಿ, that will place, this is a explant, we'll wash it with the solutions because ಏನಾದ್ರು microbes grow ಆಗಬಾರ್ದು ಅಂತ we'll wash this explant with solutions and place it on a medium containing nutrients. This is a medium containing nutrients for this tissue to grow. ಸ್ವಲ್ಪ ದಿವಸ ಆದ್ರೆ it differentiates into mass of tissues called callus. ಗ್ರೋತ್ ಆಗುತ್ತೆ, ಇಷ್ಟು ದಪ್ಪ ಗ್ರೋ ಆಗುತ್ತೆ, that is called callus. ಆ ಕ್ಯಾಲಸ್ನ ನಾವೇನಾದ್ರೂ ಬೇರೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಶಿಫ್ಟ್ ಮಾಡಿದ್ರೆ ಈ ಕ್ಯಾಲಸ್ನ ಬೇರೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಮೀಡಿಯಂ ಶಿಫ್ಟ್ ಮಾಡಿದ್ರೆ ದಟ್ ಗ್ರೋಸ್ ಇನ್ ಟು ಎಂಬ್ರಿಯೋನ ಮತ್ತೆ ಬೇರೆ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ವಿತ್ ಹಾರ್ಮೋನ್ಸ್ ಹಾಕಿದ್ರೆ ಸೈಟೋಕೈನಿನ್, ಎಸ್ಪೆಷಲಿ ಸೈಟೋಕೈನಿನ್, ಆಕ್ಸಿನ್ಸ್, ಗಿಬ್ಬರ್ಲಿನ್ಸ್, because those are plant growing hormones. If we put all these three hormones in the medium, we'll get shoots and roots. Auxins shoots, root tips grow ಆದರೆ ಗಿಬ್ಬರ್ಲಿನ್ಸ್ germination elongation, internodal elongation, cytokinin it divides and the plant grows. ಈ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಮೀಡಿಯಂ ಅಲ್ಲಿ ಗ್ರೋ ಆಗಿದ್ದ ಪ್ಲಾಂಟ್ ನ ಸಾಯಿಲ್ ಒಳಗೆ ಹಾಕಿದ್ರೆ ವಿಲ್ ಗೆಟ್ ಅ plant. This is called tissue cultured plant. ಇದನ್ನ ಯೂಶಲಿ tissue culture banana G9 ಅನ್ನೋ ವೆರೈಟಿ will develop through tissue culture, and some of the orchids, ornamental plants, ಹೂವಿನ ಗಿಡಗಳನ್ನ tissue culture ಅಲ್ಲಿ ಮಾಡ್ತೀವಿ. This is an artificial method that we are developed in the laboratory conditions. Okay, advantages ನೋಡಿದ್ವಿ ಅವಾಗಲೇ, and main disadvantages this are vegetative propagation in plants. ಏನ್ ಮಾಡುತ್ತೆ, we saw in the advantage that genetically similar ಅಂತ, there is no variation in vegetative propagation ಅಥವಾ asexual reproduction, there is not much variation. And what is the drawback? Because of this genetic similarity, that itself will become a drawback. ವೇರಿಯೇಷನ್ ಇಲ್ಲದೆ ಇರೋದ್ರಿಂದ ಅಕಸ್ಮಾತ್ ಯಾವುದಾದ್ರೂ ಒಂದು ರೋಗ ಬಂದ್ರೆ ಅಥವಾ ಒಂದು ವೆದರ್ ಕಂಡೀಷನ್ಸ್, ಎಕ್ಸ್ಟ್ರೀಮ್ ಕ್ಲೈಮ್ಯಾಟಿಕ್ ಕಂಡೀಷನ್ಸ್ ಏನಾದ್ರೂ ಬಂದ್ರೆ, ಈಗ ಬೇಕಿಲ್ದಿರೋ ಕಾಲದಲ್ಲಿ ಜೋರಾಗಿ ಮಳೆ ಬಿದ್ದು ಗಿಡ ಎಲ್ಲ ನಾಶ ಆಗ್ಬಿಟ್ರೆ ವಿ ಲೂಸ್ ಆಲ್ ಪ್ಲಾಂಟ್ ಟಿಶ್ಯೂಸ್. ನಾವು ಮತ್ತೆ ಗ್ರೋ ಮಾಡಕ್ಕೆ ನಮ್ಮ ಹತ್ರ ಏನೂ ಇರಲ್ಲ. ವೇರಿಯೇಷನ್ಸ್ ಇಲ್ಲದೆ ಇರೋದ್ರಿಂದ ದೇ can't withstand the extreme climatic conditions because there is no variation in genetics. This is the main advantage of vegetative propagation ಅಥವಾ asexual reproduction. Okay, these are the artificial methods of vegetative reproduction in plants. Previously we saw this with natural methods of vegetative propagation from stem, leaves and roots. These are the natural form of vegetative reproduction, whereas these are the artificial methods of vegetative reproduction. And now manusharu madodu idu. Okay, now after seeing all this vegetative reproduction, now let us see how sexual reproduction takes place in plants. Sexual reproduction andre we knew that two individuals are required. One is male parent and a female parent are required. Illi sexual reproduction andre fusion of male and female gametes. Gametes andre enu I'll tell. Fusion of male and female gametes leads to a production of new plant that is sexually reproduced. Usually this occur in flowering plants. Flower itself acts as a reproducing organism, an organ. Flower itself acts as a reproducing organ. Yake flower as, this is a structure of the flower, sepals, and green color sepals irutte. This is a pedicel, sepals, and colorful petals irutte. Okay, these are called petals, which will be colorful. Color varies from flower to flower, and it has a male reproductive part. This is a male reproductive part called stamen. This stamen, the tube-like structures are called filament, and at the end of the filament there is a bilobed structure, ಎರಡ್ lobe ಇರೋ structure called anther. Okay, this forms the stamen. This is a male reproductive part. In the center there is a female reproductive part called pistil. At the base there is a bulb-like structure called ovary, and it grows into a thin tube-like structure called style. At the end, there is a platform. This is called stigma. Okay, this is a female reproductive part that is present in the flower. Earlier classಲ್ಲಿ hormones ಅಲ್ಲಿ we had seen plants ಅಲ್ಲಿ sexual hormones, female reproductive part will be pollen grains, and the male reproductive part is pollen grains, whereas female reproductive part is ovary, right? This is the ovary, female reproductive part. It has ovary, style, stigma. Style is a tube that grows from the ovary, and here is a platform like structure, globe like structure called stigma, and it is sticky, ಅಂಟಂಟಾಗಿರುತ್ತೆ. ಯಾಕೆ ಅಂತ ಆಮೇಲೆ ಹೇಳ್ತೇನೆ. This is a male reproductive part, grows from the base of the ovary. This filament tube, thin tube is called filament. At the end, we have a bilobed structure called anther. On this anther, small pudi ತರ pollen grains irutte anther mele. Anther releases a pollen grains. These pollen grains have a pollen which is a male reproductive cells. Okay, pollen produce ಮಾಡುತ್ತೆ anther ಅಲ್ಲಿ. Ovary ovule reproduce. Ovule is the female reproductive cell, whereas pollen is a male reproductive cell. ಇದರಲ್ಲಿ ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ ಎರಡು ತರ ಫ್ಲವರ್ಸ್ ಇರುತ್ತೆ. plants are two types of flower. ಒಂದು unisexual flowers, ಎರಡನೇದು bisexual flowers. What is unisexual flowers? Unisexual flowers ಅಂದ್ರೆ it has male and female reproductive organs in separate flowers. ಕೆಲವೊಂದು ಪ್ಲಾಂಟ್ಸ್ ಅಲ್ಲಿ, ಎಸ್ಪೆಷಲಿ ಪಪಾಯ ಅಲ್ಲಿ, this is a male flowers present in one plant and the female flowers in one plant. ಎರಡು ಒಂದೇ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಇರಲ್ಲ ಪಪಾಯ ಅಲ್ಲಿ. ಮೇಲ್ ಫ್ಲವರ್ಸ್, ಫೀಮೇಲ್ ಫ್ಲವರ್ಸ್ ಎರಡು ಸೆಪರೇಟ್ ಗಿಡದಲ್ಲಿರುತ್ತೆ. ಓಕೆ, whereas in maize, this is a male flower, tassel ಅಂತೀವಿ, whereas cob ಅಂತೀವಿ female flower. In the same plant, ಮೇಲ್ಗಡೆ ಮೇಲ್ ಫ್ಲವರ್ ಇರುತ್ತೆ, ಕೆಳಗಡೆ female flower ಇರುತ್ತೆ. Okay, these are unisexual flowers. Here male and female flowers are present in the same plant, whereas in papaya male and female flowers are present in the different plant. Okay, whereas bisexual flowers, for example hibiscus, daswala, sasve, adralella, in the same flower female and reproductive organs are present in the same flower. ಒಂದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಮೇಲ್ ಅಂಡ್ ಫೀಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಆರ್ಗನ್ಸ್ ಇದ್ರೆ ಬೈಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್ ಅಂತೀವಿ. ಒಂದೇ ಗಿಡದಲ್ಲಿ ಬೇರೆ ಬೇರೆ ಫ್ಲವರ್ಸ್ ಇದ್ರೆ ಅಥವಾ ಬೇರೆ ಬೇರೆ ಗಿಡದಲ್ಲಿ ಬೇರೆ ಬೇರೆ ಫ್ಲವರ್ಸ್ ಇದ್ರೆ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್ ಅಂತೀವಿ. ವಾಟ್ ಲೀಡ್ಸ್ ಟು ದಿಸ್? ದೀಸ್ ಟೂ ಫ್ಲವರ್ಸ್ ಏನ್ ಮಾಡುತ್ತೆ, ಹೆಂಗೆ ರಿಪ್ರೊಡಕ್ಷನ್ ಆಗುತ್ತೆ? ಲೆಟ್ ಅಸ್ ಸಿ. This is a structure. Just concentrate on the picture. Don't read this. This is a plant with the flower, bisexual flower having female reproductive part and a male reproductive organs. ಏನಾಗುತ್ತೆ, when both pollen grains and ovary mature, ಈ ಆಂಥರ್ಸ್ ಮೇಲಿರೋ pollen grains, see this is a reproductive structures, this is a filament and this is a anther. By ಎರಡು ಲೋಬ್ ಇದೆ anther. It releases the pollen grains. ಈ pollen grains by the wind, ಗಾಳಿಯಿಂದ pollen grains come and settles here on the stigma. Transfer of these pollen grains onto the stigma of the female reproductive part is called pollination. ಈ pollen grains ಈ ಸ್ಟಿಗ್ಮಾ ಮೇಲೆ ಅಟ್ಯಾಚ್ ಆದರೆ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಪಾಲಿನೇಷನ್ ಆಗುತ್ತೆ ಅಂತ ಹೇಳ್ತೀವಿ. ಅಂದ್ರೆ ಪೋಲನ್ಸ್ ಗಾಟ್ ಸೆಟಲ್ಡ್ ಆನ್ ಟಾಪ್ ಆಫ್ ದ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಆರ್ಗನ್. ಇಲ್ಲಿ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಆರ್ಗನ್ ಮೇಲೆ ಕೂತ ತಕ್ಷಣ ಅಂದ್ರೆ ಟ್ರಾನ್ಸ್ಫರ್ ಆದ ತಕ್ಷಣ ವಾಟ್ ವಿಲ್ ಹ್ಯಾಪನ್, it will develop, pollen develops into a tube, pollen tube ಆಗಿ ಇಲ್ಲಿ ಟ್ಯೂಬ್ ಗ್ರೋತ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ ಇನ್ಸೈಡ್ ದ ಸ್ಟೈಲ್. It should enter, right? It should enter the style. To enter the style, it should need, ಅದಕ್ಕೊಂದು ದಾರಿ ಬೇಕು. So it develops a tube path called pollen tube. It pollen tube style ಒಳಗೆ ಗ್ರೋತ್ ಆಗ್ತಾ ಹೋಗುತ್ತೆ to reach the ovary. ಆ ಟ್ಯೂಬ್ ಒಳಗೆ ಪೋಲನ್ ಏನ್ ಮಾಡುತ್ತೆ, ಇಟ್ ರಿಲೀಸಸ್ ಅ ಸೆಲ್ ಕಾಲ್ಡ್ ರೀಜನರೇಟಿವ್ ಸೆಲ್. ಈ ರೀಜನರೇಟಿವ್ ಸೆಲ್ಲು ಇಟ್ ಡಿವೈಡ್ಸ್ ಇಂಟು ಟೂ. ಅಂದ್ರೆ 2n ಇದ್ದಿದ್ದು ಇಟ್ ಬಿಕಮ್ಸ್ n. 2 gametes will be formed of n, haploid nature. Okay. Pollen regenerative cell, inside the pollen tube, it divides into 2. So 2 gametes will be formed. Male gametes will pass through this pollen tube. Pollen tube ಮೂಲಕ pass ಆಗಿ it does not enter directly into the ovary here. It moves across this and enters the ovary at the micropylar end. ಇದನ್ನ micropylar end ಅಂತೀವಿ. This is a chalazal end. Usually pollen cells, pollen gametes, male gametes enter the ovary through micropylar end. ಯಾಕೆ ಮೈಕ್ರೋಪೈಲಾರ್ ಎಂಡ್ ಅಲ್ಲಿ ಗ್ರೋತ್ ಆಗುತ್ತೆ ಅಂದ್ರೆ, see in this structure, this is a structure, this is a chalazal end, this is a micropylar end. Pollen comes from through the pollen tube, male gametes come through the pollen tube and enters here because here is the egg cell. Ovary ಒಳಗೆ egg ಇರೋದು ಈ ಎಂಡ್ ಅಲ್ಲಿ. So ovary enters from this, male gametes enter from this side of the ovary. ಇಲ್ಲಿ ಎಂಟರ್ ಆದ ತಕ್ಷಣ ಎರಡು ಗ್ಯಾಮೆಟ್ ಇರುತ್ತಲ್ವಾ ಪೋಲೆನ್ ಟ್ಯೂಬ್ ಅಲ್ಲಿ, ಒಂದ್ ಗ್ಯಾಮೆಟ್ ಏನಾಗುತ್ತೆ, ಎಗ್ ಜೊತೆ ಫ್ಯೂಸ್ ಆಗುತ್ತೆ. It gets fertilized into zygote. This is called fertilization. ಇದನ್ನ one gamete ಇದ್ರ ಜೊತೆ ಫ್ಯೂಸ್ ಆದ ತಕ್ಷಣ that leads to fertilization and forms a zygote. ಅಂದ್ರೆ ಇಲ್ಲಿನೂ egg will be in a haploid nature, n ಇರುತ್ತೆ. Male gamete n ಇರುತ್ತೆ, it forms 2n. Zygote will become fertilized and form diploid, 2n ಆಗುತ್ತೆ. ಅಂದ್ರೆ 2 chromosomes ಇರುತ್ತೆ. ಇನ್ನೊಂದು ಪೋಲಾರ್ ಸೆಲ್ ಇರುತ್ತಲ್ಲ, ಮೇಲ್ ಗ್ಯಾಮೆಟ್, ಇನ್ನೊಂದು ಪೋಲನ್ ಟ್ಯೂಬ್ ಅಲ್ಲಿ ಸೆಕೆಂಡ್ ಪೋಲಾರ್ ಮೇಲ್ ಗ್ಯಾಮೆಟ್ that fuses with the centrally polar nuclei ಇರುತ್ತೆ, ಅದ್ರ ಜೊತೆ ಫ್ಯೂಸ್ ಆಗಿ this is 2 cells and the third male gamete will fuse together, form a triple fusion, triple fertilization ಆಗಿ it becomes a endosperm. ಇಲ್ಲಿ ಫ್ಯೂಸ್ ಆಗೋ male and female gamete forms zygote, whereas in the center of the ovary there is a 2 cells called polar nuclei, adru jote male gamete fuse agi endosperm form agutte. This endosperm provides food for the growing embryo, growing zygote. Okay, this provides fusion, triple fusion ago endosperm provides food for this zygote. It fuse erdu ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್ ಒಂದು ಎಗ್ ಜೊತೆ ಒಂದು ಪೋಲಾರ್ ನ್ಯೂಕ್ಲಿಯೈ ಜೊತೆ ಫ್ಯೂಸ್ ಆದ್ಮೇಲೆ ದಿಸ್ zygote after fertilization it undergoes sequence of division, successive divisions ಆಗುತ್ತೆ. ಯೂಶಲಿ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಸೆಲ್ಸ್ ಅಲ್ಲಿ ಮಿಯೋಸಿಸ್ ಆಗುತ್ತೆ, ಸೆಲ್ ಡಿವಿಷನ್, ಮಿಯೋಟಿಕ್ ಟೈಪ್ ಆಫ್ ಸೆಲ್ ಡಿವಿಷನ್, whereas vegetative part ಅಲ್ಲಿ mitotic division. Here it undergoes sequence of successive cell divisions and become embryo. Division agi it becomes embryo within the ovule. This is a ovule. Within the ovule, it becomes an embryo. Whereas the outer layer, ovary, this is a ovary, this is a ovule. Idralli, this is the ovule and this is the ovary. This will become the embryo and this will become the fruit. Embryo form agi, the outer portion, ovary becomes the fruit and the embryo becomes the seed. Ovule becomes the seed. ಹಣ್ಣೊಳಗೆ ಬೀಜ ಇರುತ್ತಲ್ಲ, that is ovule. Whereas ಹಣ್ಣು is the ovary of the female reproductive organ. Okay? Which after fruit ಆಗ್ತಿದಂಗೆ ಈ ಸ್ಯಾಪಲ್ಸು, ಪೆಟಲ್ಸು, ಆಂಥರ್ಸು ಇದೆಲ್ಲ it gets dried off. Once the fruit is formed, the other parts of the flower gets dried off. After some time, when we eat the fruit, ಈಗ ಅಕಸ್ಮಾತ್ ನಾವೇನೋ ಮ್ಯಾಂಗೋ ಒಳಗಡೆ seed ಇರುತ್ತೆ. We'll eat all the fruits and the remaining seed, after some time drying ಎಲ್ಲ ಆದ್ಮೇಲೆ at favorable condition, if we sow that seed, because of the embryo present, we'll get that seed get germinated. ಆ seed ಒಳಗೆ one baby plant ಇರುತ್ತೆ. That is called embryo. This embryo is called baby plant that will be present inside the seed. ಈ seed ನ ನಾವೇನಾದ್ರೂ ಮಣ್ಣಿಗೆ ಹಾಕಿದ್ರೆ it germinates into shoot and root, and again we get a new plant. This is a cycle of sexual reproduction in plants. Okay, first to pollination ಆಗುತ್ತೆ, transfer of pollen from male reproductive plant, that is anther, to stigma, that is a female reproductive plant. This is pollination. Once the pollination occurs, pollen tube will be formed into the style. Through this style, ಇದ್ರ ಮೂಲಕ polar cell gets divided into gametes. And the two gametes form ಆಗುತ್ತೆ, which is applied in nature. It enters through micropylar end. It enters the ovary through micropylar end, because at micropylar end, egg is there. One male gamete fuses with the egg to form zygote. Second male gamete fuses with the polar nuclei to form a endosperm. ಇದು zygote ಆದರೆ ಇದು endosperm. ಇದು 2 plus male gamete 3. So triple fertilization ಆಗುತ್ತೆ endosperm, whereas ಇದು single fertilization. Okay, this endosperm provides food for the growing zygote. Zygote later it divides successive cell divisions agi, it forms an embryo within the ovule. Embryo becomes the seed. This ovule becomes the seed and embryo becomes the baby plant inside the seed, whereas the ovary, outer layer of the ovule is the ovary, that becomes the fruit. Once we eat the fruit, we'll get the seed. At a favorable conditions, if we plant the seed again in the soil, it germinates and shoot and root development takes place, we'll get a again new plant. This is a cycle of sexual reproduction in plants. Okay, now we saw that pollination occur ಆದರೆ ಮಾತ್ರ we'll get a seed. Fertilization, pollination, fertilization, zygote formation, embryo formation, a seed ಆಗುತ್ತೆ. How this pollination? There are 2 types of pollination in plants. ಈಗ ಸೇಮ್ ಪ್ಲಾಂಟ್ ಅಲ್ಲಿ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್ ಅಲ್ಲಿ flowers will be, male flowers and female flowers will be either there in the same plant or in the different plants. ಅಕಸ್ಮಾತ್ ಈಗ see this is a female flower and this is a male flower and this is a female flower. Imagine maize. ಇಲ್ಲಿ ಮೇಲ್ಗಡೆ ಮೇಲ್ ಫ್ಲವರ್ ಇರುತ್ತೆ, ಕೆಳಗಡೆ ಫೀಮೇಲ್ ಫ್ಲವರ್ ಇರುತ್ತೆ, ಕಾಬ್. ಇಲ್ಲಿಂದ ಪೋಲನ್ ಇಲ್ಲಿಗೆ ಬಂದ್ರೆ self pollination, and because pollination occurs in the same plant. Okay, self pollination. Whereas Imagine if this is were to be hibiscus ಹಾಗೆ ಒಂದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಫಿಮೇಲ್ ಅಂಡ್ ಮೇಲ್ ರಿಪ್ರೊಡಕ್ಟಿವ್ ಆರ್ಗನ್ ಇರೋದ್ರಿಂದ ಇಲ್ಲಿ ಪೋಲನ್ ಇಲ್ಲೇ ಪಾಲಿನೇಟ್ ಆದ್ರೆ this is also self pollination. If pollination occurs in the same flower or between two flowers of the same plant, both are called self pollination. Okay, these two are called self pollination. ಅಕಸ್ಮಾತ್ one plant pollen ಇನ್ನೊಂದು ಪ್ಲಾಂಟ್ಗೆ ಹೋದ್ರೆ this is called cross pollination. Pollen from one plant, if it gets pollinated onto the stigma of the other plant, that is called cross pollination. Cross pollination occurs in unisexual plants, unisexual flowers that have male flowers and female flowers in a different plant. Okay, let us see the differences between and other parts. After pollination, the remaining cycle of the sexual reproduction remains same. Now let us see what are the differences between self-pollination and cross-pollination. Self-pollination, as we saw, transfer of pollen grains from anther to the stigma of the same flower or on other flower of the same plant. This is self-pollination. If it transfer from one plant to the stigma of the other plant of the same species, that is cross-pollination. In self-pollination, both stigma and anther mature at the same time. Self-pollination ಎರಡು ಒಟ್ಟಿಗೆ mature ಆಗುತ್ತೆ, whereas cross-pollination ಅಲ್ಲಿ it matures at a different time. And ಇದರಲ್ಲಿ self-pollination ಅಲ್ಲಿ pollen grains production will be very less, whereas here it will be more. ಇದು ಯಾವಾಗ ಬೇಕಾದ್ರೂ ಆಗ್ಬೋದು ಪಾಲಿನೇಷನ್, ಈವನ್ ವೆನ್ ದ ಫ್ಲವರ್ ಇಸ್ ಕ್ಲೋಸ್, ಬಿಕಾಸ್ ಎರಡು ಒಂದೇ ಫ್ಲವರ್ ಅಲ್ಲಿ ಇರೋದ್ರಿಂದ ಕ್ಲೋಸ್ ಆಗಿದ್ರು ಪಾಲಿನೇಷನ್ ಆಗ್ಬೋದು. Whereas in cross pollination, flower should be open for pollination to occur, and pollen grains directly transfer ಆಗುತ್ತೆ. ಇದಕ್ಕೆ ಪಾಲಿನೇಟರ್ಸ್ ಬೇಕಾಗಲ್ಲ. Without pollinators also pollination happen, because close ಆಗಿರುವಾಗಲೂ ಆಗೋದ್ರಿಂದ without any help pollination occurs in self pollination. Whereas in cross pollination ಆಗಕ್ಕೆ it requires pollinators. ಏನಂದ್ರೆ ಹನಿ ಬಿ ಅಥವಾ ಬಟರ್ಫ್ಲೈಸ್ ಅಥವಾ ವಿಂಡ್ ಅಥವಾ ಬರ್ಡ್ಸ್ ಏನ್ ಮಾಡುತ್ತೆ, ತಗೊಂಡೋಗಿ ನೆಲೋ ಕೂತ್ಕೊಳ್ಳುತ್ತೆ, ಬಟರ್ಫ್ಲೈಸ್ ಅದೆಲ್ಲ ಅದ್ರ ವಿಂಗ್ಸ್ಗೆಲ್ಲ ಲೆಗ್ಸ್ಗೆಲ್ಲ ಪೋಲನ್ ಅಂಟ್ಕೊಳ್ಳುತ್ತೆ, and it goes and sits on the another flower. Pollen grains attach ಆಗಿದ್ ವಿಂಗ್ಸ್ಗೆ ಅಥವಾ ಲೆಗ್ಸ್ಗೆ ಎಲ್ಲ ಅಟ್ಯಾಚ್ ಆಗಿರುತ್ತೆ pollen grains, that gets stuck to the stigma of the other flower. That's how pollinators help to pollinate in cross pollination. So for cross pollination, pollinators are required. We know progenies of self pollination will be genetically similar, ಇಲ್ಲಿ genetically different ಇರೋದ್ರಿಂದ we can see variation. ಇಲ್ಲಿ variation ಇರಲ್ಲ. Okay, genetic variation will not be much, whereas genetic variation will be more here. ಇದರಿಂದ inbreeding ಅಂತ ಕರಿತೀವಿ. We see this, we call this as inbreeding, and whereas ಇದು outbreeding. Inbreeding ಇದ್ರೆ ಸಪ್ರೇಷನ್ ಜಾಸ್ತಿ, ಡಿಸೀಸಸ್ ಬಂದ್ರೆ ಇಟ್ ಕೆನಾಟ್ ಟಾಲರೇಟ್ ದ ಡಿಸೀಸಸ್. ಓಕೆ, ಅಥವಾ ಸ್ಟ್ರೆಸ್, ವಾಟರ್ ಸ್ಟ್ರೆಸ್ ಅಥವಾ ಸಾಲ್ಟ್ ಸ್ಟ್ರೆಸ್, ಎನಿ ಸ್ಟ್ರೆಸ್ಸಸ್, ಇಟ್ ಕೆನಾಟ್ ಟಾಲರೇಟ್ ದ ಸ್ಟ್ರೆಸ್ಸಸ್ ಆರ್ ಎಕ್ಸ್ಟ್ರೀಮ್ ಕಂಡೀಷನ್ಸ್. ವರಸ್ ಇದು ಸನ್ಫ್ಲವರ್, ಪಪಾಯ ಇದರಲ್ಲೆಲ್ಲ ಕ್ರಾಸ್ ಪಾಲಿನೇಷನ್ ಆಗುತ್ತೆ, ದೇ ಕ್ಯಾನ್ ಟಾಲರೇಟ್ ದ ಅನ್ಫೇವರಬಲ್ ಕಂಡೀಷನ್ಸ್, ಬೇರೆ ಬೇರೆ ಕಂಡೀಷನ್ಸ್ನ ಟಾಲರೇಟ್ ಮಾಡುತ್ತೆ.